Hello, today is lesson 6. It is an anatomy lab. And if you're watching this online, two things have happened. Either we're not back in school or I've assigned this as part of your hybrid week. But this is on epithelial tissues. And if you were in the lab and doing this, I would have several boxes of slides out. And I would have showed you little pictures from using a projecting microscope of what the simple epithelial tissue looked like in the squamous tissue, cuboidal, columnar tissue, etc. And then it would have been your job to take the slides and one at a time focus in on them and identify whether it's simple squamous, stratified squamous, simple cuboidal, stratified cuboidal, pseudo-stratified columnar, which is the ones that aren't all straight, nice columns. There are some in-between pieces that are columns, but not that nice, straight, complete piece. And then you would also have been required to write down where you'd find these, whether it's in the cilia of the lungs or in the fallopian tubes, whether it's the lining of digestive organs, etc. So let's go over some of the examples. The first set of pictures here are from permission by another university. And I like to tell students that this is a university. I've taught anatomy at universities, and basic anatomy class, that first year class, is very similar to what you learn here, but just a lot more details. And so here we're looking at our slides. Let me get to the next picture. And here we have simple squamous. And the reason why I have this highlighted is a few reasons. Number one, it is this purpley pink, and that would be exactly what you'd be looking at in the lab. This is a stain. This is not what your tissue actually looks like under a microscope, but it is stained that color, so it is much easier to see. And here we have sort of a mess. It's not cubes, it's not nice columns, it's just a sheet of tissue. And this is simple squamous. And here they've shown you the darker pieces are the nucleuses. Those stain much darker. You should remember that from your CP Bio lab or your bio lab, where you took onion skin or onion and you stained it with iodine, and the nice rusty brown colored circular type objects were your nucleuses. And then they're also showing you that this is attached to underlying tissue, but this itself is not epithelial. So this is your squamous up here. And here we have a picture of simple cuboidal epithelial. And here they often are in this nice circular pattern. And this is not the best of the slides, but because it's on a low power, you're not seeing that every little piece around here is a separate cube. So here, this one is a little bit easier, where you're seeing 1, 2, 3, 4, a ring of little cubes. And in the slide that I've given you for identification as part of your lab, it's much more evident that it's cuboidal. And remember, it doesn't have to come in this circular pattern as well. You may have it in cubes in lines. And here again, they're showing you the in-between fluid is cytoplasm. They're showing you where a nucleus is. And here is something I referred to in lesson 5 lecture, the basement membrane, that generally epithelial is attached to what they call the basement membrane, that other tissue. Here we have simple columnar. It's just one row. Notice how tall it is. So very different from the cuboidal, which will be nice squares. Again, we have cytoplasm, we have nucleuses in here. This would have been a better picture here, this nucleus here, I think, but all of these are your nucleuses in the columnar tissue. Generally, that columnar tissue is very easy to identify. And again, they're showing you that that's attached to basement membrane tissue. In other words, that it's almost always that epithelial will be attached to other tissue. Here we see the stratified squamous. So first thing you have to notice is you try to identify, do I see columns or cubes? No, it's sort of a mess, and that tells you then squamous. But it is not just one single row of squamous. So here we have a mass of this, and so that is stratified because it's more than one layer. What they do show you, interestingly enough, is that although we want you to focus on this, In this particular slide, they're showing you the reality that you will have squamous tissue, basement tissue, and oftentimes columnar or cuboidal attached to that. So here they're showing you that there is some cuboidal on the edge, but in here, the slide that I give you for the squamous, it has predominantly just squamous tissue, so it'll be a little bit easier for you to identify that. And here is your pseudostratified, and this is actually in the cilia. So cilia would be, you'd be finding that in lung tissue where we need the flow of gases, and you would also find it in fallopian tubes where again the cilia is helping to push the egg through the fallopian tube. And it is pseudostratified because we have our stratified layer, but we also have other sort of mixture cells inside of that. Here we have basement membrane. They're showing you the nuclei. If I move down. And these are your examples because I'm not being able to show you the examples in the lab. So now starts your actual lab where you're going to look at the next set of slides and you're going to identify, and you don't need to just use the examples I gave you because the examples I gave you are just a few examples. There are more that you could look at. You're supposed to identify what type of tissue you believe it is, and then you need to look up and find out where this would typically be located. And then you are going to submit it. Once you've completed this, go through all of your slides and complete this, you would then turn that in onto Google Classroom under your Lesson 6. And thank you.